this spring and summer, then we can resume our face-to-face meetings.
Yeah, I know the Goldschmidt conference in July is hybrid, so you can make a choice.
Yeah, I think the Goldschmidt is hybrid, but the Gordon conference this summer, the organic geochemistry one, is live. I mean, we'll see if that continues. Hopefully. That would be my first
It's
time.
better than dead, that's for sure.
Coming out from under the rock would be my first appearance.
Under the rock or out of the mud.
Okay, it's 9 a.m. East Coast. So Peter, are you ready?
Yeah.
Okay, good morning, good afternoon, good evening. This is Paul Liu from NC State University, and welcome you again to the World River Delta System Source to Sink webinar series. And today is number 6 of this year. And this talk series is co-sponsored by NSF National Science Foundation, North Carolina State University, Louisiana State University, and East China Normal University in Shanghai, Utrecht, and CSDMS community modeling community, the modeling group. And today we are very happy to invite Professor Peter Raymond from Yale University, the Yale School of the Environment, come here talk about the hydrology and the biogeochemical controls on the watershed DOM transport. So before I introduce Peter, as always, I would like to mention our previous talk. And this is already, we have, thanks to the pandemic, we condemn the pandemic, but at the same time, pandemic give us a very good opportunity for this community can gather every week online. So we have so far have already 102 talks archived on our YouTube channel. So they are available from the YouTube or the Bilibili station. So please use it freely for the research education. As soon as you give the credit to the speaker. And at the same time, we also have a Twitter account, Source to Sink. So as you can see, the Twitter account will announce every week some talks. So welcome to follow us. And next Wednesday, same time, we have Professor Liviu Matenko from Utrecht University come here to talk about the interplay between tectonic sediment dynamic and gateway evolution in the Daniel River system. So please mark your calendar. So we know the review from Woods Hole, study the Daniel River. There's another professor also review study the Daniel. So that should be a great talk. I'm looking forward to that. So Peter, as I mentioned, is a professor from Yale University, Yale School of the Environment. And he got his PhD from WIMS, the best institute in the world, and call it William Mary. So I think Peter and I came to WIMS in 1997. So I only have maybe 2 years overlap with Peter. So then Peter got a postdoc at Woods Hole, and then 2002, assistant professor, associate, and full professor, and stay the same place. So Peter, maybe you should jump the boulder around. So but anyway, Peter's research focused on chemistry and the ecology of the inland water, and this including exchange of the greenhouse gas between inland water and the atmosphere, and the controls on the transport of terrestrial element to inland and the coastal waters. So how the storm and the drought impact that, the water system, and also use the radiocarbon to explore the age and turnover the carbon in the aquatic ecosystem. So I think Peter, now I will stop sharing. Now I'll give the floor to you. Please share your screen. And go ahead for a talk.
Thanks so much, Paul, and thanks for inviting me. Can you see the slides?
Yes, yes. Very
All right.
good.
I'm just going to get a laser pointer going. Okay, yeah, so I thought what I'd do here is sort of discuss this pulse-shunt concept that myself, Jim Sayers, and Bill Sobczak published in 2016. Sort of introduce folks to the concept, go through sort of the conceptual model we came up with, and then end showing some more recent data that's sort of exploring the pulse-shunt concept with actual data. Just as a reminder about dissolved organic matter, right? I think the best analogy for DOM is that it's watershed tea. So you pour rainwater on a watershed. It interacts with any stock of organic matter, being the live leaves, the leaf litter, the soil organic matter, and dissolves organic matter into the water. It gives it that sort of brown color, at least in terrestrial systems. We like to call it a master variable because it performs a lot of functions and impacts a lot of different ecosystem processes. I've listed a bunch here. It absorbs light, and so it can compete for light. It also absorbs UV light, which can be a positive thing because UV light is just as harmful to aquatic organisms as it is to us. It's like one of the main transport mechanisms for pollutants. So like my lab just did, an exercise of trying to determine the amount of mercury coming out of rivers. And, to do that, you have to know how much DOM and POM is coming out. It's an energy source to bacteria. It has nitrogen and phosphorus associated with it. It can impact water treatment because during different points in water treatment, you can form harmful disinfection byproducts from the DOM. And one of the interesting things and reasons why this is a fun variable to study is that all of these different processes that we're listing here, like the composition of DOM helps determine how important it is for these different processes. So, we all know it's this wide mixture of different types of compounds and components, and the composition that you have will determine how much heavy metals are bound, how good of an energy source it is, etc. And so, the talk that I'm going to give is trying to come up with like a conceptual idea for how DOM behaves at a whole drainage network scale. And so to do this, right, you're building off of 40 years of scientists trying to do drainage network level scaling. This is not an easy thing to do, mostly because getting the measurements to sort of ground your conceptual model is difficult. It's difficult to sample an entire, say, 6th or 7th order drainage network. Nonetheless, there's lots of concepts out there to build off that, and it arguably sort of starts or really kicks off with the river continuum concept, which most people have probably seen. I'll just check. Is the chat for me, Paul, or anyway? The river continuum concept did a lot of things, but it had some focus on organic matter, and it kind of stated that downstream communities sort of work off the organic matter that was already reworked by upstream communities. So sort of downstream communities relied on the inefficiency of upstream communities, and it created this continuum of both particulate organic matter and DOM at the network scale. Relevant for this talk, it also sort of recognized how the physical nature of these systems change as you move from small stream orders that are light limited, particularly in temperate systems where you have shading. And as you move downstream, your canopy opens up. You can get to the large river where you have enough light, where you can start to support, and you have enough depth where you can start to support almost these lentic-like ecosystems, right, with phytoplankton and zooplankton. And so that's also could be important for something like DOM, the switching from more of like a heterotrophic system with a PR ratio of less than 1 to an autotrophic system, maybe as you move downstream. The RCN actually, directly talked about DOM. It called it this soluble organic carbon, and it said, you'd get this very sort of diverse set of compounds come in across a terrestrial aquatic interface. It would start to get reworked, creating even more diverse set of compounds, due to microbial action. But then as you move downstream to larger and larger stream orders, the diversity would decrease as you're sort of removing large amounts of DOM as a whole. So there's a lot of other conceptual papers on DOM. Another one that's sort of received a lot of attention is looking at discharge concentration curves as you move from small first order streams to larger rivers. And if you look across a lot of systems in general, what you start to see is that DOC increases with discharge in these small streams, and I'm really going to show a couple of nice examples of that. But as you move to larger and larger rivers, the behavior becomes more chemostatic, like down here, where the concentration doesn't change with flow. And there's a lot of potential reasons for that. And I'm going to sort of come back to this towards the end of the talk and show one example of why we think this is happening. But I mean, the pulse-shunt concept is pretty simple. It basically works off of a couple premises. The first is that hydrologic events, that is an increase in discharge, lead to or often accompanied by an increase in concentration. And so a lot of the terrestrial DOM delivery occurs during these low frequency hydrologic events. And I'll walk through that in more detail. The other part is that if you are DOM being transported during these low frequency high discharge events, your residence time in the drainage network goes down. So that's the shunt. So you pulse in with the hydrologic event, but because it's a hydrologic event, sort of by definition, your residence time in the network is lower, leading to less percentage uptake during transport, just due to sort of a time constraint on processes removing the DOM. So let's go over this in some detail. This is some of the earlier evidence showing an increase in DOC concentration with a hydrologic event from Liz Boyer. It's from the Deer Creek in Colorado. This is a snowmelt hydrologic event. So you can see, you're building your snowpack over the winter. The snow starts to melt, leading to high discharge. And if you look at DOC, right, concentrations are low in the stream all through winter. And the interesting thing is when discharge goes up, the concentration responds by also increasing, right? And since flux is concentration times discharge, this is that pulse of DOM. Here's another example from Hinton, some small catchments in Ontario around Harp Lake. These are fall precipitation events for 3 different watersheds. You can see the hydrograph here, right? In these fall precipitation events, so these punctuated discharge events. And the same thing in here, where in general, right, you see an increase in concentration with these hydrologic events, not dilution. And so we published a paper in 10,010, Jim Sayers and I, where we sort of did a metadata analysis, right, where we looked for small watersheds, less than 60 kilometers squared. We tried to exclude watersheds that had a significant wetland contribution, and we were, looking for watersheds that had a decent number of measurements. So back then, 5,000 measurements in a metadata analysis was pretty good. And basically, we found that this behavior was, generally stood for these watersheds across eastern or maybe northeastern US, where on average, the response was an increase in concentration with discharge. because we were doing these at weird sites, we were able to quantify sort of, the impact. And basically, one of the numbers we put out there was that 60% of the DOM export occurs during, like 15 days of the year. So this is that pulse. And it's relevant from like a mass transfer point of view, but it also has implications, with respect to the way we sample these systems, because, it's hard to chase storms. But if you're interested in the DOM sort of being put into these drainage networks heading towards the coast, you really have to target them to get a representative sample. This paper and others had found that you get a higher jump in concentration if it hasn't rained in a while, although it's not huge. And that also you usually get a higher jump in concentration if it's warmer. And this second one was demonstrated very nicely by Henry Wilson, who was a postdoc working in the Harvard forest. So these are a bunch of ISCO captured events, small stream, forested stream, where you could see we're seeing this, discharge concentration curves with an increase throughout the whole year, but they become more pronounced in the warmer months, and maybe even most pronounced in the fall. And so, there's a lot of potential reasons, and we can maybe discuss this later, of why you would think you have higher concentrations in warmer months. Part of it could be hydrologic reasons, other sort of biogeochemical reasons. But this has also been shown more since then, this sort of temperature influence on the QC curve. But I'll be using some of this response type curve when we build the model. And the pulse, it's pretty simple. Like I said, we're just hypothesizing that there's a relationship between the discharge and residence time at the drainage network scale. And I'll walk through how we tried to treat that. But, these are really nice examples of some of the systems we sampled. And these are events. And so you can see, large amounts of water coming over these weirs, and you can see the brown color of this terrestrial DOM sort of pulsing into the drainage network. And the theory for why this happens is, been around for a while. The basic idea is that during a hydrologic event, the water table rises, and you get flow of water that goes through the mineral poor upper layer of the soil directly to the stream. So it's bypassing the potential for the DOM to sort of move through this mineral layer that almost acts like a chromatograph, slowing the DOC down, the DOC zorbing and desorbing. And so if you can bypass that and leach this sort of fresher DOM straight to the stream or river, you get this higher concentration with Q. And part of the idea is, there's a couple different ideas, but one of them that's intuitive is that the larger and larger the discharge event, the more and more of this upper layer gets connected directly to your stream or river. And then the shunt, like I said, all we're sort of saying here is that there's some relationship between discharge, Q or discharge, and the network residence time. This is something that's actually kind of really hard to work out, residence time at the network scale. It's not an easy thing to do, but I think it's intuitive or hard to argue against the fact that there's some relationship between discharge and network residence time. And basically what we're saying is that when residence time is long, there's more chance for DOM reactions and removal than when residence time is short. So kind of going from the active pipe during short residence time to a passive pipe during, active pipe during short residence time, passive pipe during longer residence time. This has also been articulated probably better mathematically by Will Walheim in this paper. A bunch of us published more recently. where basically what you're trying to work out is, the ratio of supply of DOM, which is a function of discharge and concentration, and the demand, which stream scientists will sometimes write as a function of the width, the length, and the width, the length, and the depth. And so, kind of what we're also saying here, actually, it's the width, the length, and the reaction rate. U is the reaction rate. I'm sorry, it's the reaction rate in meters squared. And so, kind of what we're saying here is that with respect to these DOM dynamics, that the flow frequency curves can matter, and I'll show some examples of this. So, if you have most of your flow coming out with moderate size events 1 year, you might have much different dynamics and different DOM delivery to the coast than if you have a year that's dominated by a couple of really large hydrologic events. So we've become very interested in like how flow frequency varies within watersheds. So we built a simple model to demonstrate this. We built what we thought was a representative New England watershed that's 8th order. We determined the travel time of water using network scaling laws and hydrography data. I'll show that. We then assigned a decomposition constant to DOM. This is kind of tricky and somewhat pulling a number out of a hat. And then we used this to assess reactivity of terrestrial DOM. But basically, as you'll see, when we get to a stream order, you can rewrite those equations that Will had. And these might be more familiar to folks more like thinking about diagenesis, where if you know the length and velocity in your stream length, right, this sets the residence time, and you're multiplying it by a decomposition constant to get the fraction used. So you can rewrite and so this is how my brain thought, right, being a marine organic geochemist, but it's the same way that Will wrote, just sort of rearranging the equation. And so you heavily rely on, this stream order of drainage networks. And for those of you who don't remember, the way this works is you have your smallest streams are first order, right? And you have when two first order streams meet, you get a second order. When two second order streams meet, you get a third order, so on and so on. Scientists since the 20s really have realized that these drainage networks are fractal in nature. That's kind of what this diagram is demonstrated from Strahler's paper. So that scaling laws drop out of this, such as the scaling law of stream length. And the way this works is if you know the ratio of the length of the first order to second order, say this is 1 kilometer long and this is 2 kilometer long, that ratio will hold as you move up in stream order, right? So this one will be 4 kilometers long, this one will be 8 kilometers long, and so on and so on. There are similar scaling laws for the area of watersheds, the number of streams and rivers. And so nowadays you can gather these up for your watershed, right? There are products available that you can gather these up and sort of use these in these types of scaling laws. And we did this for Connecticut, and you can see how some of these scaling laws work out, like the number of stream orders, right, decreased by about, the next order has about 25% of the streams as the preceding order, and in particular river length, which I already said we're going to use, right, changes by about a factor of 2 as you move up in stream order. But we set these ultimately, we went and queried what New England looks like, and then we set these scaling laws for our model. And then the last thing you need is this Tukanuga constants. This is Japanese scientists in the 60s who also use these scaling laws to allow you to get the connectivity. And so basically the way this works is you can set up a Tukanuga table, right? And it tells you the number of first order streams that are connected to a second order, number of first order that are connected to a third order, right? The number of third order that are connected to a seventh order, and so on. And it's basically, this equation is two constants. One is this 2.7, and the other one is moving up, how this ratio changes moving up in rows. And this is something you can also sort of gather now from hydraulic products like NHD+. And this is the Tucanaga table for Connecticut. And so then once we have this, you can imagine it's even like Excel spreadsheet, right? And so what the model does is it takes these CQ curves to pump DOM into the headwater streams. And so, what we're creating is a model to like really just focus on that terrestrial organic matter. And then you look at the uptake, following that equation I just showed, where you need to have a time, and we use length over velocity, and a decomposition constant. So velocity, now you can also sort of dig out from these hydraulic exponent equations that folks are exploring sort of in earnest now that we have big data. And these are sort of countrywide or basin-wide relationships between discharge and velocity. So if you assign a discharge, right, you can get a velocity for different stream orders, right? And so this is a table where we're looking at the velocity for a discharge event, which is about 0.8 centimeters per day, which for the Connecticut is sort of the discharge that does the most work for moving DOM into the drainage network. And we can talk about that if people want me to spell that out a little bit more. So you can see that as you go up in stream order, right, you're predicting an increase in velocity, which is what happens. But you're also, the length scale is also going up. So if our residence time is length divided by velocity, right, you can see that the length scale increases faster than the velocity scale. So what we're predicting, and what others have sort of predicted also, is that the residence time is higher as you move up in stream order. And again, that's just because the length scale increases faster than the velocity scale. And these are some other papers that have sort of demonstrated the same thing. And this is partly arguing for, a renewed interest in bigger stream orders, rivers, for removal processes. The decomposition constant, right? This is a tricky one. It's hard to get a decomposition constant for DOM in any ecosystem. This is not any different for streams and rivers. There were a couple out there. And so we, picked one and then assigned Q10 to it, so we could have it change as a function of temperature. But, the choice of a decomposition constant is obviously pretty important, and it would be great if we knew more about it. And so basically what we found is we came up with this diagram, which is the number is the stream order, and the size of the circle is the amount of removal, for an average year in this eighth order model temperate watershed. And so indeed, we found the largest removals, mass uptake occurs in the largest systems. The lines represent the amount of this uptake that came from the stream order that the line is coming from. So we also basically find that it's really only the preceding 3 stream orders that are flowing into that 8th order system that, basically bring enough DOM in to matter. And so there was some idea that maybe small headwater streams are important, even to large rivers, for bringing in really fresh new DOM. And it's hard to argue that from just the fact that their mass transport, the volume of water at DOM that they add is so small compared to these preceding 3 stream orders. And so we kind of came up with this law of stream subsidies, right? You can actually work out the fraction of DOM removed in the 8th order that's coming from the preceding stream orders. And so just 2 thought exercises here using the model, and then I'll show some new data. One is we asked the question, well, what if you had a watershed that had 60 centimeters of discharge a year, but those discharge were delivered in different size events, going from one watershed where you had 60 events of 1 centimeter, so this is a small rain event, or 12 events with 5 centimeters. This is a decent rainfall, at least around here, or 6 really large events. And you can see that the flux from land goes up if you believe those QC curves, where concentration goes up with discharge event. So even though the volume of water is the same, this is sort of that concentration increase response. The mass uptake goes up just simply because you're moving more DOM in, but the fraction removed goes down. And if the amount goes up and the fraction removed goes down, right, this has perhaps the most interesting implications to the flux to the coast, right, where they're going up almost by a factor of 4 compared to an increase in discharge, increase in the flux off land, maybe a factor of 2.5. So they sort of multiply together, the pulse and the shunt, to lead to large coastal inputs. The other thing we did is we held the size of the event constant. So this is the results for a discharge event of about 2.5 centimeters. We used the Q10 of 2 and then asks what happens during different seasons. So basically here we're asking, you get an increase in concentration, which is greater at higher temperatures, and you get an increase in uptake also at higher temperatures, and how do these two sort of balance out at the network scale? So here you can see that increase in the amount delivered due to the change in the QC curve. Again, you can see changes in the uptake due to the Q10 effect that we assigned. But interestingly here, what we find is that the uptake outpaces the increase in delivery with increase in temperature. So the warmest temperature is actually the smallest decrease to the coast. And it's somewhere in the middle where you get optimized exports. And we thought this was interesting, particularly thinking about the fall, when temperatures are around 15 degrees and leaf litter drops, and we often see the biggest increases in concentrations, sort of emphasizing their potential importance. All right, I should be good to be done in like 10-15 minutes. So, we did get some funding to look at this, and we focused on the Connecticut River locally, partly because we're chasing storms. And so what we did is the Connecticut River is kind of cool, where it's this long, depending on how you count it, 6th or 7th order river, right, with all these 4th and 5th watersheds stacked up on it, moving from a pretty big climate gradient, at least temperature, right, where up here you have like snowmelt dominated watersheds and down here you don't. We sampled a number of the tributaries, these yellow dots, sort of synoptically, and then these 2 watersheds with the, I guess, orange dots. We put out ISCOs and we put out ISCOs and sort of FDOM probes and SONs to really look at them in detail. And we've started to publish quite a bit on this. And you can get an idea. These are the high intensity sampling watersheds. We had a pretty good range in watershed area, going from a tiny watershed up in Pope Brook to the Connecticut River itself. So in fact, the USGS had an FDOM probe out at the Thompsonville, so almost further south site that you can get before you hit tidal water. We looked at that data in detail. And so here is just a graph showing the QC curve, where the C here is FDOM from the probe, and how the QC curve looks in the winter, spring versus the summer. And FDOM is kind of nice because it is maybe arguably focusing on that terrestrial end member, that humic-rich DOM. And so we saw this behavior at the mouth of the Connecticut, right? And we were able to sort of see these dynamics that we postulated in the pulse-shunt at the mouth of the Connecticut, where we also saw like some of these temperature effects, and we described in detail in this paper by Matt Schultz, who was a master's student. Jake Hosen was a postdoc in the project and published a bunch of papers. One with the ISCOs, right, we're doing a lot of this sort of what I call high throughput organic geochemistry, right? These EAMs and UV scans of the DOM, which gives you some fingerprinting capacity. And we use these to build the end member mixing model to look at the allochthonous DOM and the autochthonous DOM coming out of that same site that Matt had looked at. So this is at the mouth of the river. And so interestingly, I really like this graph, is what we're showing here is QC plots on the top, and then Q flux plots on the bottom. And if you focus in on just the total DOC concentration, we saw this chemostatic behavior, right? So DOC concentrations didn't vary with discharge much at the mouth of the Connecticut, exactly as predicted in Irina Creed's paper. Interestingly, though, when we pulled apart through our mixing model the allochthonous and autochthonous components, we did see an increase in concentration of this sort of terrestrial allochthonous component with discharge. And we saw the opposite, or a decrease in the autochthonous component with discharge. And so this holds for the reasons I just showed, you know, even in this graph here, right? If you try to focus in through DM quality measurements on the terrestrial component, you clearly see this increase in concentration at this site with flow. And we could do this here also with our end member mixing model. We also saw it. This we've published on other papers where we really see that this temperate watershed doesn't have really high rates of GPP or autochthony until you get to low flow conditions where the residence time increases and some of the suspended sediment drop out and the DOC concentrations are low. All of these, you know, finally provide the favorable light conditions, and the water temperature is warm for high growth. And so, you know, basically what we say is that for the Connecticut River, this chemostasis is maintained by source switching, going from allochthonous material at high flow to autochthonous material at low flow. When you start to think about the fluxes, though, because the allochthonous dominates at high flow, I forget the exact number, but I think like 70% of the DOM coming out of the Connecticut was allochthonous because its concentration is higher when flow is higher. Jake went further and sort of did an analysis of how DOM quality changes with residence time across the US with a large range in watershed size. So we did travel time in a very similar way that I just explained for the conceptual pulse-shunt model. And he found a pretty good relationship between some of these DOM indices, like the freshness index and the spectral slope ratio, and residence time, right? Land cover didn't really matter much. Even temperature was hard to find an effect. Residence time provides this dominant signal. And if you elongate residence time by having, a big lake or reservoir right upstream of where you're sampling, these relationships get even more pronounced. So it really suggests that the downstream shifts in DOM are driven by these in-stream processes that need time to develop. All right, just 2 more slides. And so we also have spent some time looking particularly at photooxidation as a potential removal process and how effective it is in this watershed. This graph is a little hard for me to step through, but it's basically looking at the amount of DOM that's coming out of the Connecticut River that is still photolabile. We did this through incubation experiments, and we concluded that about 50% of the DOC exported from the Connecticut is photolabile. This is work by Brian Yoon, who's a PhD student. And so the conclusion here was sort of, we inversely argued that if you have a large amount of photolabile DOM coming out of the river, and particularly at higher flows, that this process is not efficient enough to remove significant amounts of DOM. Some of the reviewers didn't like this inverse sort of approach, so we also, Taylor Mavara, PhD student, built a model for the Connecticut, which we published, where she took sort of the proper chemical approach, right? We're modeling wave light input to the system, light attenuation, absorption of radiation by DOM, and even the quantum year. Well, we modeled this at the network scale, so all the different stream reaches. We did this for different flow frequency, where here's a really low event, right? With a Q98, using a Q98. Here we find, even at the sort of lower end of flow, that we can only, the model predicts about a 10% removal. Where if you get down to flows that are maybe more meaningful for mass transport, like median flow or a Q15, you're getting very low percentage removals of DOM predicted from this modeling approach. And it would predict having large amounts of photolabile DOM being exported from the Connecticut River, which is what we see. So that's it. This is my last slide. And so the conceptual diagram that we sort of ended with is there was this big paper by John Coles arguing that systems are an active pipe, not a passive pipe. So rivers don't simply shunt terrestrial materials to the ocean, but they're sites of very active processing. We are sort of arguing a nuance where you move from active to passive as a function of discharge, right? Where as discharge goes up, you become more passive. And the important thing here is, if you're interested in fluxes to the coast, particularly for DOM, since the concentration goes up with flow, the passive nature is important for determining how much is getting to the coast. So that's what I had for a talk, Paul. I'll take questions.
Peter, this is a fantastic, it's a very, very interesting talk. I did learn a lot. So let's see, we have many audience here, so let's open to them. So if you have any question, please go ahead, unmute yourself, just ask yourself. I say it's Hans Paul, unmute. Hans, you have any question?
Well, first of all, hi, Pete.
Hey, Hans.
Hope you're doing good.
Yeah.
I know you're familiar with this, but this increase that we're seeing in DOM and DOC in our coastal zone, do you think that's purely due to the fact that storms are just becoming more intense, and that the ability of the stream system to remove the DOM DOC has gone down? according, just exactly as the way you explained it in your last several slides.
Yeah, I think that's one factor, right? But there are other arguments for getting just an overall increase in DOM, linked to like changes in sulfate delivery to watersheds. So it's definitely a part. It might be interacting with some of these other sort of big level processes, but yeah, I think it's definitely a factor.
Yeah, and the big problem we're having is separating that from human disturbance, of course, in the watershed itself.
Yeah, it's tricky working at the watershed scale.
Especially when you're not a person who has a lot of expertise in it. Well, yeah. I see Tom Bianchi's got his hand up, so I'll give the floor to him.
Okay, nice seeing you, Hans.
Thanks, Hans. Very good talk, Pete. I guess this whole idea of the pulse shunt, it depends on where you're shunting to, right? I mean, it seems like a lot of the connection between the upper watershed soils, if you look at, increased frequency of storms, and you say maybe the storage or export from those soils will decrease, because the replenishment rate, if you keep increasing the storm, you don't replenish it. But the whole connection with those soils and what ultimately gets to the ocean gets interfered with, because you're not really shunting to the ocean. It's only those last 3 orders of streams that are impacting what's making it to the coast. So I guess I'm losing the connection between the really upstream soils and replenishment to this, how things kind of get occluded along the way, and where the soil to coast matters, how far up you have to go. I don't know if that.
No, I get it. I mean, the antecedent effect has been demonstrated a bunch before, right? So if you have a storm that's right on the heels of another storm, you do get a depression in the QC curve, but it's pretty slight, right? And so the way I think about it is there's definitely, some loss in the stock of DOM available to be transported. But in general, it was like, I forget, but it was like a 2% decrease in our metadata analysis. So I think in general, like it's not much transport limit. There's a little transport limitation, but it's pretty small. And then, with respect to the shunt, like for the Connecticut, we're saying, here, let me bring up that slide. But for the Connecticut, we're saying, yeah, it's the 4th and 5th orders that are important for bringing DOM in. But that's all these big watersheds flanking the river, right? And so the first order streams way up here are still adding DOM that's going in, traveling through the 4th and 5th orders. And so they're still important. It's just these first orders that are right on the river that I'm saying aren't important. If that makes sense.
Yeah, no, that I see that. And then if I could just squeeze one more thing in, Pete.
Sure.
Is as you start getting into the tidal or salinity effect, the salt wedge, and this may be outside of your area of interest, I'm just wondering if you think there's a difference in the flocculation or potential removal of the allochthonous versus the autochthonous DOC in terms of what ultimately makes it. Do you see what I'm saying?
Yeah. I mean, the Connecticut would be super interesting because it has like a long stretch of fresh tidal. So your longest residence times there are really like in that zone. And so that's probably like a really important zone. And then, like our estimates of how much photolabile and stuff that's being delivered to the coast sort of match with some of the estimates of like how much of a river's terrestrial DOM people think are photooxidized in the coastal zone too. So I think they're like, once you get there and the sediment drops out and the light field gets nice, I mean, it's probably a really important site for removing this stuff. I'm not exactly sure about flocculation. That's one that's always puzzled me. I mean, it's definitely happening, but I'm not sure to what degree.
Thank you.
Yep.
That's great. Anybody else? So that's waiting, when we're waiting other people prepare the question. Peter, you know, this is a totally outside my expertise, you know, my understanding, but I do have a follow the Tom the question about the frequency and the intensity of the storm, the impact to the DOM. So your sampling is just a one-time sampling, or you have a repeat sampling?
Yeah, we have these ISCO things, which we're able to, like, you see a storm coming, you tell it to sample over the whole storm. Yeah, so you try to get, like, yeah, a bunch of samples over the storm.
Do you have a comparison for a little bit historical, like a, you know, a dry year, wet year, different season, you know?
Yeah, we had that some in the metadata analysis, and so that's where we saw, like, this clear temperature effect. So you like Henry Wilson showed at the Harvard Forest, and we even saw at the mouth of the Connecticut. There's clearly a temperature effect, and like part of that could be, you know, in a watershed like the Connecticut, you have frozen soils.
Yeah.
In the cold months, so you know your flow paths are different with temperature, but then you also have, you know, less GPP in the watershed. You know, you don't have the leaf drop happens, but that stuff starts to get leaf decomposed, and so there's some mixture of hydrology and biogeochemistry that are important there, and that's what we need to work out partly.
Yeah, the one thing maybe, you know, to think about, for example, the winter, during the winter, how much snow, you know, the amount of snow, because this will also the temperature, as you mentioned, and this will affect during the spring, how much melting water, how much discharge.
Right.
Will be interesting correlation.
Yeah.
Okay, we have two more. Bob, yeah. Bob, go ahead, and then Christoph. Yeah.
Yeah, I'm wondering about the interaction between episodic event and or events and the delivery and movement of the solid material, solid carbon material in the stream bed, and also the correlation with the reactivity of the DOM as a function of time after the event. You used a single K for most of your considerations, and I'm just wondering if in fact there's a spectrum of Ks that varies with events.
Yeah, so for the first question, you know, we collected a lot of filters in our sampling, and we just haven't had the time to run them. So we have all these event POC data that we haven't had to run, but I think the shunt is more important for POC. I mean, I'm sorry, the pulse, right? The concentrations, a few people have shown that the concentrations go up even more than DOC for POC. The problem is, as you know, like trying to get travel times of POC, how long it takes that particle to get out the Connecticut River is, you can't treat it as simply as we did for DOM. So it's outside my area of expertise, because you have it potentially settling out and moving. And so maybe the shunt isn't as big a deal. I don't know. And then the decomposition constant, there has been some recent work that shows intuitively that the decomposition constant goes down as you remove DOM from the pool. And so I think that would, you know, be something you could now add to a model, and Taylor, who's doing some following up model, is sort of doing that, where the decomposition constant is related to residence time and basically goes down as residence time goes up. But then, you know, I mean, in some of our incubations, and, you know, this is up Tom's alley, is like, if you add some photooxidation in there, like, who knows what happens, right? Then you might be creating this really labile stuff, you know, through the whole network. And so the decomposition constant is an interesting but tricky one. It's just hard to measure.
Right. Thanks.
Yep.
Okay, Christoph.
Yeah, well, I guess my question also regarding the particulate part of the carbon flux. And yeah, well, I think you answered to the question in your previous answer, but do you have a comparison for the Connecticut, for example, for how much during these pulse events come as a particulate phase compared to the dissolve phase in POM, DOM?
iDOPA Sriamidar has done some work kind of close, and the POC increase, I'm blanking on the size of the watersheds he was looking at, but the POC increase is more pronounced. You get a bigger increase in concentration in the POC than the DOC. This is the way I remember his papers.
Okay, thanks.
Okay. Anybody else?
Hey, Pete, does the pulse shunt work in the Arctic?
I think it works best up there, right? There are these papers, Max Holm had this thing where he did biolability throughout the year. And like the spring up there is when you have maximum pulse and shunt, and you see like real high amounts of really labile DOM coming out the river, which is sort of an inverse argument, right? That it's not being used. And so I think it's probably pretty big. Yeah.
Okay, almost 10 o'clock here. So if you know any further question, I think we thanks Peter again. And this talk, once again, this talk is archived on our YouTube channel. If you want your student or your research group to rewatch again, please go ahead to go there. You can watch. And otherwise, if no other question, we can end it here. Thank you very much for Peter and all the audience. I'll see you next week or in the near future.